So the HDL molecules are synthesized in the liver and also in the intestine, and from the intestine or from the origin of synthesis, they will be moving, they will be released into the circulation. And once this nascent HDL will be discoid in shape, they will be oval in shape, and their size will be too small because they are not having any lipids in them. They will be only occupied with the apoprotein, ApoA1, ApoE, and ApoC, and they slowly move towards the target side of the peripheral tissues. And during the course of their journey, what happens? They come and meet across all these molecules, all the other lipoproteins and all, and they finally reaches the peripheral tissues. And on reaching the peripheral tissues, this HDL comes and gets bound onto the peripheral tissues. The cells, they get attached onto, they get fixed onto the cells with the help of the receptor. And this receptor we call it as scavenger receptor B1 or SRB1. That is the receptor we have seen there. Yeah, this is known as scavenger receptor B1. So this is a receptor present on the cells onto which the HDL gets attached. Right, so the HDL molecules with the help of the apoprotein, only the apoprotein part is attached onto the receptors, right? So that's one of the functions of the apoproteins. They enables the binding of the lipoproteins onto the target cell through the receptors. So the receptor is actually interacting with the apoproteins, not onto the lipid part or the lipoproteins, right? The apoprotein part is attached onto the, is getting attached onto the receptors. So the HDL molecule with the help of the apoproteins binds onto the cells through the receptors known as scavenger receptor B1 or SRB1. So these receptors are present in all the cells of the body, including the hepatocytes or the liver cells, right? So this SRB1 on the peripheral tissues or on the target tissues, or when the HDL, nascent HDL comes and gets attached onto the peripheral tissues, this SRB1 will lock the HDL onto it. And what does this SRB1 do? It enables the entry of the cholesterol esters from the cells into the HDL molecule. So that is how the HDL is laden with the cholesterol esters. So again, the HDL molecule is having ApoA1 on their surface. So this ApoA1 activates the enzyme LCAT. So as a result of this activation of this enzyme, what happens? Lecithin releases its phospholipid, it's this polyunsaturated fatty acid PIFA on the second carbon atom of lecithin, and it is accepted by the cholesterol. So this cholesterol becomes cholesterol esters. These cholesterol esters will now get entered into the HDL molecule, right? So that slowly what happens, the disc-shaped HDL start getting converted into spherical molecule because the HDL is getting accommodated with the cholesterol esters, right? And this HDL molecules which are now spherical in shape now comes out. They will be released back into the circulation and they will start move towards the liver, because the function of HDL is to take the cholesterol esters from the peripheral tissues to the liver, right? So they comes out of the peripheral, or they gets detached from the peripheral tissues and enters into the blood, and through the blood or through the systemic circulation, now they moves back to the liver. And during again the course of their journey, what happens, this HDL will be coming across all the other lipoproteins, VLDL, IDL, LDL, എല്ലാം ഈ വഴിക്ക് ഇങ്ങനെ പോയിക്കൊണ്ടിരിക്കുന്നുണ്ട്, right? So what does HDL do? HDL with their cholesterol esters, what happens when they come across the other lipoproteins, these cholesterol esters will be slowly released to the other lipoproteins along with ApoC and ApoE. So that again the cholesterol esters, the concentration of the quantity of the cholesterol esters will be getting reduced in the HDL. So these when these cholesterol esters are given to the other lipoproteins, what does they do? That is VLDL, IDL, LDL, etc. The LDL, etc. will be having small quantities of TG, while VLDL is having large quantities of TG. So they will be exchanging the TG with the cholesterol esters. So cholesterol esters will be released to the VLDL, IDL, LDL, while the VLDL, IDL, LDL will be releasing, or they will be giving TG in return to the HDL. So now this HDL with cholesterol esters and small quantities of TG, they are known as HDL2. So HDL actually belongs to different subclassifications, subtypes are there, HDL 1, 2, 3, etc. there. So HDL2 is that HDL which is having cholesterol esters, along with them they will be having TG. Right, while HDL1 will be having only free cholesterol in them. Sorry, not free cholesterol, cholesterol esters will be there. So HDL molecules are synthesized in the liver and also the small intestine. They will be having higher protein content, but when immediately secreted from the liver and the intestine, they are known as nascent HDL. They will be having no cholesterol or cholesterol esters. They will be having cholesterol and cholesterol esters only when they reach the peripheral tissues. So they are having other they are having the apoproteins, ApoA1, ApoE, and ApoC, right? And these carries out very important functions, while ApoA1 is having the major role, that is they activate the LCAT, and hence they act as anti-atherogenic in nature. And what does they do? LCAT breaks down the lecithin molecule, or they will be releasing PUFA from their second carbon atom, and it is estrifying the cholesterol, so that free cholesterol is now converted into cholesterol esters, and they will be scavenged from the peripheral tissues or extrahepatic tissues, and they will be taken towards the liver. And from the idea and chylomicron producing cholesterol esters that is dissolved with the HDL core. Okay. So this is the reaction happening. That is LCAT carries out the conversion of lecithin to lysolecithin and cholesterol to cholesterol esters. And what happens? These cholesterol HDL2 now move towards the liver, and on reaching the liver, what has to be done? This cholesterol has to be delivered, right? So again, on the liver cells also, what is there? SRB1 is there, scavenger receptor B1 receptors are there. So these receptors, what does they do? These receptors now takes up only the cholesterol, right? While HDL and ApoA1 will be taken back, or they will be released back onto the there will be they will be remaining outside the hepatocytes. Only the cholesterol esters will be taken up by the liver cells. So this is a function of SRB. So SRB is having two different functions. In the liver cells, what does it do? They accept the cholesterol esters from the HDL without internalizing HDL molecule. While in the peripheral tissues or the extrahepatic tissues, what are the SRB1 doing? They internalize the HDL molecule, and will be these HDL molecules will be laden up with the cholesterol esters. So they are having dual functions. In the liver, they are accepting the cholesterol, while the extrahepatic, in the hepatocytes, they are accepting the cholesterol esters from the HDL molecule without internalizing the HDL or the apoproteins. Only the cholesterol esters are extracted out from the HDL molecule. While in the case of extrahepatic tissues, the molecule, the HDL molecule, will be internalized first, and they will be provided with the cholesterol esters, right? So the initial HDL molecule, nascent HDL, is discoidal in shape, and it's known as pre-beta-1 HDL or nascent HDL. And what is happening? They will be slowly laden up with ApoA1, the ApoC, ApoE, etc. And what happens? They carries out the efflux of cholesterol or cholesterol transfer protein, right? Cholesterol ester transfer protein, or a cholesterol efflux regulatory protein, is enabling the transfer of cholesterol, right? And it is taken up by the HDL molecule. And as the cholesterol content increases, the HDL particles become spherical, and further, they are enlarged by accepting more and more free cholesterol, which in turn is esterified by LCAT, which is known as HDL2. Now, this HDL2, as the cholesterol ester content of the HDL2 increases, the cholesterol esters begin to be exchanged by TG. By the other lipoproteins. So this is known as the HDL2, and they are finally delivered to the liver with the help of scavenger receptor B1. Okay, and HDL molecule will be coming out. They will be recirculated. So this is how the metabolism of HDL take place, right? So this is a VLDL and this is a HDL. The VLDL is rich in TG. They are having small amounts of cholesterol esters. So what is happening? The TG will be donated to the HDL, while cholesterol esters are taken up by the VLDL. So this is happening with the help of cholesterol ester transfer protein, CETP. Okay, so this is the liver and small intestine. They synthesizes a discoid shape nasal HDL. This nascent HDL, what from the peripheral tissues, the free cholesterol, etc., will be esterified, and they will be forming HDL3 first. This HDL3 then increases in their size by adding more and more quantities of cholesterol esters, and they will be released into circulation. And these cholesterol esters will be transferred to the VLDL, LDL, IDL, etc. And in exchange, TG will be accepted by them, and they finally move towards the liver. And what happens in the liver? In the liver, with the help of the SRB1, the cholesterol esters are free cholesterol and cholesterol esters will be liberated or will be released from the HDL molecule, and they will be secreted into the bile. So the final thing happening is the secretion of cholesterol into the bile, right? So that is how the metabolism happens, right? So the subfractions of HDL are HDL 1, 2, and 3, and again 3 is again subdivided to 3A, 3B, 3C, etc. Right? Now, what's the clinical significance of HDL? The clinical significance is that they are inversely proportional. That is, as the level of the HDL in the serum increases, the protective action also increases, or there is reduced chance for the myocardial infarction or cardiac arrest because they are anti-atherogenic in nature. Right? So increases the HDL value should be always greater than 60 milligram percentage. If a person is having a HDL value greater than 60 milligram percentage, we can assume that the person is having a low risk of myocardial infarction, while a value of HDL less than 35 milligram percentage is at an increased risk of MI. Okay, so that is why HDL is known as good cholesterol. Now, reverse cholesterol transport, we have already studied, that is cholesterol efflux or the esterification of cholesterol in the presence of LCAT, which is being transferred from the peripheral tissues to the liver, is known as the reverse cholesterol transport, which is made possible through HDL2 and in presence of the scavenger receptor B1. So this is the reverse cholesterol, that is liver synthesizes, then discard HDL is there. Discard HDL will be now moving into the peripheral, moving towards the peripheral tissues, collects all the cholesterol and cholesterol esters, and they will be taken into the liver back, and it is this cholesterol and cholesterol esters will be finally secreted in the bile, right? So this is known as reverse cholesterol transport. This is a formation and fate of lipoproteins. Liver synthesizes VLDL, while intestine synthesizes chylomicrons. They enters into circulation. Chylomicrons actually enters into the lymphatic system, and from the thoracic duct they enters the systemic circulation. So they will be delivering the TG to the cells. So the source is different. In case of VLDL, it is an endogenous TG, while chylomicrons transfers the exogenous TG. So in presence of lipoprotein lipase, they are finally the TG will be delivered to the cells, and the VLDL will be converted into LDL. What does LDL do? LDL, again, majority of the LDL is synthesized from the chylomicrons and not chylomicrons, from the VLDL, and a small fraction can be synthesized from the liver also. So the LDL is formed. LDL transports the cholesterol molecule, cholesterol esters to the cells, right? And again, from the excess of the cholesterol will be transferred from the extra hepatic tissues to the liver through the HDL, right? And this HDL, so this is known as the reverse cholesterol transport, right? That is uptake through receptor mediated endocytosis. The cholesterol transferred from the liver to the cells is carried out by LDL, while HDL carries the transport of the cholesterol esters from the cells to the liver. So this is known as a reverse cholesterol transport, which is happening with the help of LCAT and scavenger receptor proteins. The LPA is already over. Now, what are the clinical significance of lipoprotein metabolism? What happens if any of these lipoproteins found to be impaired, especially the protective ones? So we know that the cholesterol and cholesterol esters, it is to be deposited in the fatty in the adipose tissue, and excess of the TG and cholesterol will be taken back to the liver, right? So the TG will be deposited in the adipose tissue. Only very small quantities of TG will be deposited in the liver. But what happens if the dietary intake or the endogenous synthesis of these lipids increases above or beyond the limit? What happens? It results in a condition known as fatty liver. So we might have heard about the term fatty liver. Fatty liver, from the term itself, we can understand fatty liver means liver laden with fat. So when does the thing happen? When does the fatty liver occur? Whenever there is an excess abnormal deposition of or abnormal consumption of fat happens, the body has to conserve this fat. So the fat will be deposited naturally in the peripheral tissues. Again, the peripheral tissues has got some limit. So when the limit is extended or exceeded, what happens? This fat will be taken back to the liver, and the liver will be the storage site, right?